This course in human physiology has been an exciting journey. I still remember during the first class we spoke about the phenomena of homeostasis. Indeed, we are grateful to the great philosopher and scientist called Claude Bernard. For the first time, he came up with the concept. He said that for life to survive anywhere, the constancy of the internal medium is a basic requirement, and he called it as the milieu interior. Eventually, however, in the year 1926, Sir Walter Cannon, a professor at Harvard, coined the term homeostasis, which is prevalent till today. He essentially says that the physiological systems function to maintain or regulate the condition of internal environment within relatively narrow limits. Now, what do you mean by that? We mean we can consider any parameter in the cell, outside the cell, and we try to look at the limits within which the parameter must be regulated. For example, you can think of sodium ion concentration in the extracellular medium, which has to be around 140 millioccurrents per liter. Similarly, you can think of potassium, calcium, magnesium. You can think of any of these criteria, and you have to be sure that within the extracellular medium they are within a range, and within intracellular medium they are within a different range, and that they have to be carefully regulated in the respective ranges. We have different organ systems. We can also look at those organ systems as contributing to the maintenance of homeostasis. What do you mean by that? Let's just think of lungs. Lungs are very important and essential for making sure that the level of oxygen in the blood is always kept constant. Similarly, they keep on removing the carbon dioxide and ensuring that the level of carbon dioxide in the blood is constant. You can take the example of another organ, for example, gut. We take food and the food is eventually digested and the constituents are taken to the blood. That makes sure that the components like glucose and amino acids are always constant within the medium. You can take another example of kidney. The kidney ensures that the ingredients in the blood like urea is always kept under very narrow limits. So, all these organ systems, on one hand they depend on the constancy of the medium that is provided by different mechanisms, and also they contribute to some of those elements which they themselves regulate. So, in this way you can think of temperature, blood pressure, osmolality, energy supply, they are all the different. So, how does the body know that a particular parameter has to be within a particular range? We call that mechanism as a set point. It is an abstract concept that tells us as to how, if we take the example of temperature, how the thermoreceptor mechanisms in the brain, they consist of several circuits which ensure that if the temperature goes up, the circuits are excited so that the body initiates the mechanism that lower the temperature, or if the circuits note, the changes in the circuit note that the temperature has fallen, then the mechanisms are set in operation to raise the body temperature. Now, this does not mean that there are neurons at a set temperature. No, there are circuits which respond to different temperatures, and from that the rest of the body calibrates its mechanisms for temperature control. I think then we moved on, if I remember, we moved on to understanding as to what the muscles really are. Muscles are very interesting organ. They perform a range of mechanical functions. We have skeletal muscles that enable us to move from one place to another. We have cardiac muscles that continuously run the heart, and we have smooth muscles which are lining our visceral organs like the alimentary canal. The muscles essentially consist of actin myosin filaments, and those actin myosin filaments are anchored, actin filaments are anchored on what you call as Z membrane. And you remember we talked about the sarcomere, which are lined by the Z disc, and anchored on the Z disc are the actin filaments, and they alternate with the myosin filaments, and they provide a certain geometry in which the actin and myosin filaments can slide into one another and facilitate the contraction of the entire muscle. We also recorded that the muscles are divided into dark band and the light bands, and they are essentially constituted of the actin and myosin filaments. And then there are of course the transverse tubules which take the action potential from the surface whenever the muscle cell is excited because of a neurotransmitter called acetylcholine. It can take the action potential, release calcium ions, and in the presence of calcium ions, the actin myosin filaments, they can slide at one another and bring about the contraction. The stimulus that is required for the skeletal muscle essentially comes from the nervous system. It is by way of a motor fiber that has a neuron which is there in the central nervous system, either brain or spinal cord. It is a fiber that uses acetylcholine as a neurotransmitter, and whenever acetylcholine is released on the muscle fiber, the muscle fiber which is equipped with nicotinic fibers is it opens the certain what you call as the sodium ion channels, and the muscle cell depolarizes. That facilitates the influx of calcium ions, and that leads to the contraction of the muscles. We also read something very interesting. We noted that the things do go wrong, and that gives rise to certain diseases, and the example was a very interesting autoimmune disease which we called as myasthenia gravis, in which the antibodies are generated against the receptor for acetylcholine, and as a result of that the neurotransmitter does not really go to the muscle, and the muscles become extremely weak. Then we switched on to understanding the cardiac muscle. At the histology level, we find that the cardiac muscle is very similar to that of the skeletal muscle. Here we have the Z membranes, the light bands and the dark bands, but there are some differences, very clear differences. Here in the cardiac muscles show what you call as the gap junctions, which permit the ions to travel from one cell to another cell, and thereby the cells can contract in a certain rhythm, in a certain synchrony, and then we find that the excitation, the property of the heart muscle is to excite on its own, and that happens because there is what they call as a conduction system which initiates at the level of the AV node, SA node, and that SA node when it initiates the signal that travels over the internodal discs to the AV node, and then over the Purkinje fibers to the different parts of the ventricle, and as it goes, it opens the calcium ion channels in the plasma membrane. The calcium ion channels act on release the calcium from the endoplasmic reticulum, and sudden surge of calcium brings about the sliding of the actin and myosin filaments. Whenever the heart goes into systole, the blood is pumped from the ventricles into the aorta, and from the aorta it goes into the rest of the organs. The rest of the organs, the blood is returned via the venous system to the right auricle, from the right auricle to the right ventricle, and then it is pumped to the lungs for oxygenation. We also had some numbers to quantify the amount of the functioning of the heart. For example, we said that the end diastolic volume is about 110 to 120 ml, and systolic volume means when the ventricle goes into systole, the left ventricle may pump out about 70 ml of blood, and 40 to 50 ml of blood may still be there. And if you just trace the activity of heart for a minute or so, you will find that about 5 to 6 liters of blood is pumped out, but that is under resting conditions. Under conditions of hyperactivity, the cardiac output may indeed go up by 3 or 4 folds. Then another interesting thing that we find is that whereas the blood that is being pumped out of the left ventricle is under very high pressure, the mean pressure may be about 95 to 100 mmHg, whereas the blood that is pumped from the right ventricle which goes towards the pulmonary circuit, this blood is deliberately kept under much lower pressure, that may be about 10 or 15, the mean pressure may be about 10 or 15 mmHg, and the differences are obvious because the pulmonary circuit is relatively short. You need to keep the low pressure so as to make sure that there is no leakage of plasma from the capillaries in the lungs, whereas in the case of the in the systemic system, in the systemic circuit, you have to maintain the high pressure because the blood has to go throughout the body into the distant organs. When it comes to pumping of heart, there is something very particularly the notes are very interesting because they are self-excitatory, and that happens because there's a thing called as there are some of the channels in the notes which depolarize when the plasma is hyperpolarized. They suddenly discharge the current, the channels open, and as a result of that the calcium ions go in, and in this way the rhythmic phenomena is sustained. The heart also shows a very interesting phenomena, what you called as the Frank-Starling mechanism. It really means it is the ability of the heart to adapt to the changing volume of inflow blood, which means that if the inflow of blood in the ventricle is more, then the ventricles will contract to the same extent such that whatever blood has entered into the left ventricle will be pumped out. This is to ensure that there is no stagnation of blood into the heart, and this phenomena is called as the Frank-Starling mechanism. Although the heart generates its own rhythm, it is secondarily under the control of the autonomous nervous system. There are parasympathetic fibers which control which can excite the heart, increase the rate and force with which the heart can contract. On the other hand, the heart is equipped with a nerve supply from the parasympathetic nervous system, which can reduce the rate of the rate at which the heart keeps on beating, or the frequency of the heartbeat. It is very interesting to note that Sir James W. Black came up with the molecule which we call as propranolol, which is commonly called as the beta blocker, and which inhibits the adrenergic, the beta adrenergic receptors, and they play a key role in the treatment of the angina pectoris and similar diseases. One of the easy ways to record the contractions of the heart is to apply the technique which we called as ECG or electrocardiogram. And that electrocardiogram is non-invasive, it is very useful. Every time the heart undertakes initiates a signal from the SA node, you can get the electrical signals recorded from the periphery, and any disturbance in them can be recorded and visually analyzed. Presuming that a 70 kilogram healthy individual has a volume of about 5 liters of blood, and total quantity of blood is 5 liters, and with every time the heart goes into systole, about 5000 ml of blood is pumped out. All of that returns, some of it goes to the brain, coronary circulation, kidney, gastrointestinal tract, skeletal muscle and the skin. But all these parameters are variable depending on the condition or depending on the organ that is under use. The blood, as it flows through the capillaries, a small quantity of that also is perfused out, that is generally plasma. It does not contain large protein molecules and certainly no RBCs, and these fluids give rise to the lymph, which is drained via the lymphatic system back to the venous system close to the heart. As the blood travels to the lymphatic vessels, it encounters lymphatic nodes which play a very important role in the immune system. We note that whereas the arteries are relatively thick and they can put up with lot of pressure, the veins are relatively far more compliant, and they are equipped with valves so as to make sure that the blood does not flow backwards, particularly under the influence of gravity. The blood that is in the aorta is pulsating within the range of 80 to 120 mmHg. However, the blood, as it goes towards the capillaries, the pressure has fallen to about 30 mmHg. The blood under capillaries is under very low pressure, and then it goes to the heart, and from the heart as it goes to the pulmonary, it is oscillating between 10 to 25 mmHg. August Krog one of the doyens of physiology, came up with a very interesting observation. He observed that not all capillaries are active at the same time. He showed that some of those organs which are particularly active organs and they need more blood during those circumstances, there is a increased flow of blood through the capillaries. However, the capillaries, by virtue of the smooth muscles right at the origin, they can constrict and they can reduce or retard the flow. So, depending on the organs, depending on the activity undertaken by the organ, the capillaries can open and close and regulate the blood supply. However, it is to be noted that while most of the capillaries in most of the organs, the endothelial cells show the opening through which the blood can flow out and the molecules can easily flow, particularly the small molecules can easily flow out. The capillaries in the case of brain are highly protected by way of astrocyte processes which do not allow the plasma to easily flow out, and even the endothelial cells show what you call as the tight junctions, and this phenomena we call as the blood brain barrier. This is very necessary to regulate the transport of material from the blood into the neurons or into the brain. The regulation, the flow of blood through different vessels is tightly regulated by a range of agents. Some of them are locally generated, and some of them serve as hormones. One such very powerful agent is the nitric oxide. Nitric oxide is a gaseous molecule that is released by the endothelial cells, and it can just travel across from the endothelial cells into the smooth muscles of the blood vessel, and there it can trigger the mechanisms by which the blood vessel can relax and facilitate the flow of blood. Then there are some hormonal systems about which we studied, and one such hormonal system is which is very well studied and which is extremely powerful is the angiotensin II, which comes from the action of a hormone released by kidney called as renin. The renin acts on a molecule called angiotensinogen, which is released by liver, which is eventually converted into angiotensin II. Angiotensin II is a small molecule made up of about 8 amino acids, and that can act on the blood vessels, that stimulates the secretion of aldosterone from the adrenal gland, and it stimulates the Thirds mechanism, and all put together it helps to increase the blood pressure. It is very effective, particularly when there is loss of blood or fall in the blood because of any other reason. The blood pressure is also continuously being monitored. The brain continuously monitors the blood pressure by way of what you call as the baroreceptors, which are located in the aorta and also in the carotids. If you remember, then we moved on to understanding of the function of the kidney. We recorded that the kidney is made up of different of thousands and thousands of neurons, and each nephron is made up of the Bowman's capsule that has glomeruli, a proximal convoluted tubule, a descending tubule, loop of Henle, the thick ascending tubule, the distal convoluted tubule, and eventually it goes into the collecting tubule and the collecting duct. And as a result of ultrafiltration at the level of Bowman's capsule, the ultrafiltrate travels down the nephron, and it is processed. A lot of material that is filtered but useful to the body is being reabsorbed. Molecules like sodium, molecules like glucose, they are all being absorbed at the level of the proximal collecting tubule itself, and then the ultrafiltrate travels into the distal convoluted. At every step it undergoes a change, as it travels, the ultrafiltrate is being processed until you get the formation of the final urine. Particularly the distal part of the tubule and the distal duct play a key role in making sure that the water content of the urine is very tightly regulated, meaning thereby that if there is too much excessive consumption of water, then the dilute urine will be formed, whereas if there is dehydration of the body, then the concentrated urine will be formed, and the urine can be the osmolality of urine can be as high as that of 1200 to 1400 milliosmoles. This is very high when we compare to that of the blood, where the osmolality of the blood is about 300 milliosmoles. We also studied about the structure, we studied about the podocytes at the level of the Bowman's capsule. We also learnt about the importance of creatinine. If you remember, creatinine is a molecule, is a molecule that's a breakdown of creatine phosphate, and that creatine molecule is is, uh, is to be thrown out. Any accumulation of creatinine in the blood is an indication of malfunctioning of the kidney. One of the very interesting, since the functioning of the kidney is essentially based on the blood pressure, and it's about at about 60 mmHg that the blood is being filtered at the level of Bowman's capsule. It's very necessary that the changes in the blood pressure elsewhere in the body do not impact the pressure at which the blood is being filtered at the level of Bowman's capsule. Therefore, the kidney has mechanisms by which the blood pressure is kept constant, and we call it as the autoregulatory mechanisms by which the glomerular filtration rate and the renal blood flow are fairly kept constant, although the changes may be effective elsewhere in the body. Another interesting point that we learnt about the kidney is its capability to secrete a very interesting molecule called erythropoietin, and in case there is a fall in the oxygen level of the blood, then the kidney is capable of generating this hormone which we call as erythropoietin, and that erythropoietin can stimulate the formation of blood at the level of the bone marrow, and thereby try to increase the capacity of the blood to carry the oxygen to the different tissues. We noticed that the osmolality of the fluids in the kidney, although in the kidney it is about 300 milliosmoles, we find that as we go deeper into the medulla, the osmolality of the biological fluid goes on increasing. This is necessary to make, this is essential to ensure that the kidney can generate highly concentrated urine, particularly into the circumstances when the body is being dehydrated. And in that respect, the hormone that plays a key role is the antidiuretic hormone. So, if you are being dehydrated, then the hypothalamus releases this hormone, which is released via the posterior pituitary gland, and as that hormone comes into the blood, it will act on the collecting duct of the pituitary gland of the kidney, and there it will ensure the reabsorption of water, so that the kidney can form relatively concentrated low volume urine, thereby reducing the loss of water through the urine. If you remember, then we moved on to the anatomy and physiology of the respiratory system. We learnt about the lungs, we learnt about how the lungs are organized in the thoracic cavity, guarded by the diaphragm, and how the movement of the diaphragm plays a key role in the inspiration and expiration process of inspiration and expiration. We saw how we have the trachea and the bronchi branch, and then finally they end up into the alveoli, and every time there is negative pressure generated into the thoracic cavity, the air flows in via the nose and the trachea, and the air flows into the alveoli, and in the wall of the alveoli the gaseous exchange happens, where the oxygen from the air transfers into the blood, and the carbon dioxide from the blood goes into the alveoli. And then we learned something about the partial pressures. We learned that the partial pressure of oxygen by the time the air reaches into the alveoli is about 100 mmHg. Then it goes into the oxygen. So, the partial pressure of oxygen in the blood is also about so much. And then we also learned the importance of the cell type 1, alveolar cell type 1, which lines the alveoli, and then we also studied something about the cell type 2, which play an important role in secretion of the surfactants, which form a thin lining across the layer of the watery fluid that lines the alveolar cells. This is necessary to reduce the surface tension. If you remember, if this layer of phospholipids is not there, and if the surface tension is not reduced, then the alveoli just collapse, and then no gaseous exchange is possible. So, we need to make sure that the alveoli remain open, and then they facilitate the gaseous exchange. We also learned something about the movement of the diaphragm. The diaphragm are being supplied by the phrenic nerve. Every time the impulse comes from the phrenic nerve, which brings the impulse from the medulla via the spinal cord to the diaphragm. The diaphragm expands. It generates negative pressure in the thoracic cavity that sucks in the air via the nostrils into the lungs, and then eventually the expiration starts, where the diaphragm again moves upwards. It assumes a dome shape, that is because of the elasticity of the diaphragm, and the air is pumped out of the thoracic cavity. The lungs, there is a positive pressure on the lungs, and the air is expelled. The partial pressures, the study of partial pressures is very important in understanding how the gaseous of the and try to understand how the gases travel. We noted that the at sea level, the partial pressure of oxygen is about 160 mmHg. By the time the air enters into the alveoli, it is 100 mmHg. By the time the blood passes from the lungs into the heart, there is shunt blood. As a result of that, the partial pressure is reduced to about 95 or 96 mmHg, and then that is the partial pressure with which the blood travels to all the organs. And in the muscles, as it travels to the muscles through the capillaries, the partial pressure in the tissue outside is very low. It's about maybe about 40 mmHg. So, the oxygen readily travels from the capillaries into the tissues. The tissues take up that oxygen. The tissues at the same time have higher concentration of carbon dioxide, which may be about 46 or 45 mmHg, and then it travels back from the cells into the capillaries, which take up the oxygen, and then it travels back to the lungs for gaseous exchange. Then we read something about hemoglobin, the wonderful molecule which is made up of four subunits, 2 alpha units and 2 beta units, and it shows the capability of readily combining with oxygen, converting into oxyhemoglobin. We also noted that hemoglobin molecule is made up of a porphyrin ring. At the center lies the iron atom, and that iron atom can combine with can reversibly combine with the oxygen molecule, and whereas 100 grams of blood has the capacity of carrying about 20 ml of oxygen. If it carries 20 ml of oxygen, we can say that it is fully saturated, fully oxygenated, and as the fully saturated blood from which has picked up maximum oxygen, as it travels through the lungs, it goes to the different organs, and there it gives up about 5 ml of blood. So, its partial pressure, which was about 100 at the level of lungs, falls to about 75, and the blood, as it goes away from the tissue, say muscle or any other tissue that has used oxygen, it still has about 75 ml of oxygen, and it still has partial pressure. It can still exert partial pressure about 40 mmHg. So, the point here to remember is that even the deoxygenated blood has still a lot of oxygen, which is about which can almost say 100 ml of blood is still carrying about 50 ml of oxygen, but that is very necessary that in case of there is exercise or any vigorous muscular activity, then you still have a buffer amount of oxygen which can be used under emergency cases. An interesting property of hemoglobin is that if the pH of the blood becomes slightly more alkaline, it can bind with more oxygen. If it becomes slightly acidic, its affinity for oxygen is reduced. This phenomenon was described by Bohr, and the phenomenon is called as Bohr effect, and this plays a key role. So, this is very interesting because as the blood is traveling through a tissue, suddenly it encounters a lot of carbon dioxide. As a result of that, carbon dioxide interacts with water. In the presence of carbonic anhydrase, you generate H ions, and as the H ions combine with hemoglobin, the hemoglobin's affinity for oxygen is reduced. It readily gives away oxygen, which is now available to the tissues, and exactly opposite happens by the time the blood travels through the lungs, where its affinity for carbon dioxide goes down and its affinity for oxygen goes up, so that in the lungs hemoglobin can now absorb lot more oxygen. So, this elasticity which the hemoglobin molecules shows, it really it greatly increases the efficiency of oxygen of hemoglobin to carry oxygen from the lungs to the tissues and back from the tissues to the lungs. Yet another interesting property of hemoglobin is that its affinity for oxygen is reduced when if the temperature goes up. So, supposing you do exercise and as a result of that the body temperature literally goes up by 1, 2 or 2.5 degrees, then that is because you are doing exercise and your body needs more oxygen. In these circumstances, you find that as the temperature goes up, the affinity for hemoglobin in the tissues goes down, and therefore, the oxygen can more readily release the oxygen. Hemoglobin also shows yet another interesting property, what you call as cooperativity. We have just now seen that hemoglobin has 4 subunits, and oxygen combines with the first subunit. Its affinity increases in the case of second, more in the third and more in the fourth, and this cooperativity is also plays a great role in efficiency of oxygen, in efficiency that hemoglobin shows in carrying the oxygen. Then we learned something about the smooth muscles. We found how the smooth muscles are quite different from the skeletal muscles. Several of them show connectivity by way of gap junctions through which the ions can travel and the cells can show arrhythmic activity. We found that the cells they do not have Z membrane as in the case of skeletal muscles, but they have what they call as the dense bodies. On the dense bodies, the actin molecules are anchored, and then there are myosin filaments, but the sliding of the actin myosin filaments is very much similar, and they can and we find that they can show a great deal of contraction, which is even much more efficient than that of the skeletal muscles. However, we found that the cellular mechanisms are somewhat different. In the case of skeletal muscle, we have troponin, here we have colmodulin, and we have different steps by which the skeletal muscle can show. Then we moved on to the digestion. We studied about the esophagus, we studied about the stomach, we studied about the different cell types in the stomach. If you remember, we talked about the oxyntic cells or the parietal cells which secrete the hydrochloric acid, and then we learned something about the parietal cells, their capability to secrete the hydrochloric acid as well as intrinsic factors which play key role in the absorption of vitamin B. Then we also studied about the G cells which secrete about the gastrin, and then we elaborated about how the parietal cells secrete the hydrochloric acid into the lumen. We studied that, and when we studied about the pepsinogen, we studied about how the stomach is coated with a layer of mucus which plays a great role in protecting the stomach from the high acidity in the stomach. And then we read about the importance of the gastrin, and then we talked about how the duodenum plays a key role by way of bicarbonate ions. It neutralizes the acidic chyme that is arriving from the stomach, and then how the pancreatic juices play a key role in digesting in further digesting the food and making it available for the absorption. And then we also talked about the hepatic portal system and its role in carrying the digested food from the intestine to the liver. And then we gave a considerable focus on the liver and its role in synthesizing bile, the anatomy of the liver, how they are plates of the cells which on one hand secrete bile, and on another hand it's a conduit for the blood from the portal system and from the arteries, and how the blood flows then into the central vein, and from the central vein the blood is carried to the heart. We talked about the function of bile in the digestion of the food, particularly the emulsification of the fat and the absorption of the fat, and then how the how carbohydrates finally convert into glucose are absorbed, galactose is absorbed, and how the fatty acids because of the action of the bile, we have the formation of the micelles, and how the micelles generally help in the absorption of fatty acids across the wall of the intestine. Then we moved on to study of some very interesting aspect. We studied about the hormones. We read about how some of the important interesting endocrine glands. We studied about the role of the hypothalamus in controlling the pituitary gland, how the pituitary gland is organized into the anterior and the posterior, how the anterior endocrine gland is a source for such important hormones like, if you remember, LH and FSH are the two known as gonadotropins, and the third was the TSH, which plays a very important role in stimulating the thyroid gland. And then yet another very important hormone was ACTH, adrenocorticotropic hormone. The adrenocorticotropic hormone plays a role in stimulating the adrenal cortex. And then we also talked at length about the growth hormone. We said that growth hormone comes from the pituitary gland and it has a profound effect on the growth of a large number of tissues, and also it is a great anabolic hormone. It promotes the growth of bones, growth of muscles, and growth of a large number of and then we also studied as to that there are some instances where growth hormone has a direct effect, but growth hormone also has an effect indirectly via the insulin like growth factor. Then we also read about the different hormones, how they have different half-lives, whereas amines have a half-life of few seconds. Thyroid hormone has a, we talked about T3 and T4, and we also talked about polypeptides, and then protein hormones, and how the steroid hormones have a relatively a very long. Then we talked about how some of the hormones, whereas most of the peptide hormones do not need a carrier in them, but some of the hormones like thyroid hormones has a carrier which is a huge protein molecule which is called as thyroxine binding globulin, and how the half-life of the hormone is being influenced by this binding affinity. We saw how the T4, how the T3 and T4 are being synthesized at the level of thyroid. We saw how the thyroid is made up of the thyroid follicles. Thyroid follicles is made up of single layer of cells. It secretes what you call as a colloid, and the colloid is a store, which is actually essentially made up of amino acids, which is a polymer of tyrosine amino acids. But then tyrosine is iodinated, and if a molecule of tyrosine receives and tyrosine is eventually converted what you call as monoiodotyrosine, diiodotyrosine, and then eventually into thyronine, which we call as the 3 iodide atoms. We have called this at T3 or triiodothyronine or tetraiodothyronine, which is same as thyroxine. And these hormones, when excited by the TSH, which is coming from the pituitary, thyroid will release T4 and T3, and T4 can circulate bound to thyroxine binding globulin. Mostly T3 is on its own, it doesn't bind, but eventually T3 will find its way into the tissues and it will act. T4 will also be released eventually in small proportions. It will also go, but before T4 acts, it is invariably found to lose one of the iodine, the T4 is converted into T3, and then it will act on its receptor. And we also understood the profound importance of T3 and T4 in what they call as the metabolic. So it's a main metabolic hormone. If in the absence of T3 and T4, the rate at which the metabolism is being undertaken almost falls to more than half. So it's a very important metabolic hormone. We also saw that the thyroid is a source for yet another hormone which we call, if you remember, there is another hormone which we call as calcitonin, which is secreted by the parafollicular cells of the thyroid, and it plays a key role in reducing the calcium in the blood level. What are the factors that control the release of thyroid hormone? Thyroid hormone is controlled itself is under the control of the pituitary hormone TSH. TSH itself is under the control of what you call as TRH. TRH is a small tripeptide hormone that is synthesized by certain neurons of the hypothalamus. This tripeptide is delivered from the hypothalamus via the median eminence to the pituitary gland, and in the anterior pituitary gland it acts on the TSH cells. They excite the TSH cells. The TSH cells release the TSH hormone, and the TSH hormone then acts on the thyroid gland, and in return the thyroid gland releases the hormone T3 and T4, and it is these hormones which have negative feedback effect at the level of the pituitary and also at the level of hypothalamus. So in this way the T3 and T4 regulate their own secretion by way of negative feedback effect at the level of pituitary as well as the hypothalamus. In the case of thyroid, if you remember, we also learnt about this interesting disease in which is called as a goiter, thyroid goiter, which is again an autoimmune disease. Then we spent quite some time on this very interesting gland which we call as the adrenal gland, which is a source for the steroid hormones. It has some very interesting steroid hormones like the aldosterone. We said that the thyroid gland is divided into the three components, the zona glomerulosa, the zona fasciculata and the reticularis. The outermost zone, zona glomerulosa, is particularly plays a key role in the synthesis and release of a hormone called as aldosterone. If you remember, we talked about aldosterone being under the control of angiotensin II. So whenever there is fall either in the volume of the blood or in the there is a reduction in the sodium ion concentration in the blood, then there is angiotensin II comes into the play, and then angiotensin II has a profound effect in stimulating the cells of the zona glomerulosa, the outermost layer of the adrenal cortex, and those cells of the adrenal cortex release a hormone which we said just now is aldosterone, and then aldosterone goes back to the kidney and it acts on the principal cells of the kidney, and then it plays a key role in the reabsorption of the sodium ions. So for the homeostasis of sodium ions, and sodium ions are so important for the functioning of the brain and the neurons. They play a so sodium ion concentration is very important, and therefore they need to be the amount of sodium ion concentration has to be very tightly regulated, and aldosterone plays a key role. If you remember, if the concentration of sodium ions in the blood goes up, then we have another hormone that comes into play. We talked about that also, and that hormone was called as ANP, or atrial natriuretic peptide. Then we spoke about the inner two layers of the adrenal cortex, the cortisol, the zona fasciculata and the zona reticularis. These are known to secrete mainly the hormones which fall into categories what you call as glucocorticoids, and the other was androgen like hormones. The glucocorticoids, one of the most important glucocorticoids that you get in the humans is the cortisol. It plays a key role in the carbohydrate metabolism, it acts on the liver, it plays a key role in the gluconeogenesis, that is generation of glucose from the non-carbohydrate source. Yet another very key function of cortisol is in stress. Whenever there is stress, cortisol is released and it suppresses the immune system, it suppresses the immune response, and thereby it plays a role in what you call as it accomplishes its function as an anti-inflammatory agent. Interesting to note here is that there are a lot of synthetic steroids which are commercially used or which are clinically used to treat inflammation. Yet another interesting aspect of this hormone was that it suppresses the immune system, and particularly in the cases where you need to undertake organ transplant, and where you need to ensure that there is no organ rejection. The agents similar to cortisol or glucocorticoids, they are used as immunosuppressants. I think then we also spoke about the importance of testosterone, which is a hormone which is produced by the Leydig cells of the testes. It is responsible for the generation of the maleness or the male character. It is a great anabolic hormone, and it is circulated in very high concentrations, particularly on the arrival of the pituitary. However, its concentration falls with the advance of the age. I think these were some of the topics that we covered in our course on human physiology. There are some additional topics. I hope you have enjoyed the course. We have attempted to touch upon the molecular mechanisms on one hand, and also try to correlate them with the way the different processes coordinate with one another, and finally how they can give rise to or express certain behaviors depending on the challenges that are being experienced by our systems or by our physiology. Thank you very much.